Buenas tardes. Bienvenidos a todos los que están aquí presentes y a los que nos acompañan por Zoom y por el canal de YouTube al seminario de Frontiers de hoy. Me da muchísimo gusto presentarles al doctor Wolfgang Busch. Él obtuvo una maestría en biología en la Universidad de Tübingen en el 2004. Después realizó su doctorado también en Tübingen, pero ahora en el Max Planck Institute. Del 2004 al 2008 y realizó su estancia postdoctoral en la Universidad de Durham en Estados Unidos, siempre desde entonces trabajando con Arabidopsis y plantas, desarrollo de plantas. En el 2011 formó su primer grupo de investigación en el del Institute for Molecular Plant Biology en Austria. Y en el 2017 el Salk le hizo una oferta que no podía rechazar y movió todo su grupo de investigación al Salk Institute en California. Desde entonces ha trabajado allá, fue primero profesor asociado y en el 2020 ya fue profesor del Salk Institute. Y bueno, evidentemente ha desarrollado importantes contribuciones al entendimiento del desarrollo de las raíces en plantas. Y solamente quiero resaltar dos reconocimientos que ha recibido en los últimos años y que son que fue recientemente nombrado Hess Chair de Plant Science en el Salk Institute y que junto con 4 investigadores del Salk en el 2019 con Joan Corey, Joel Noel, Julie Lowe y John Ecker obtuvieron un gran de $85 millones para tratar de desarrollar plantas que tengan la capacidad de capturar dióxido de carbono para mejorar el clima. Es algo muy relevante, que es prácticamente en lo que están trabajando ahora su grupo de investigación. Y solo me queda mencionar que Wolfgang ha publicado alrededor de 61 artículos científicos, 14 revisiones, y ha recibido alrededor de 10,000 citas a su trabajo. También ha dirigido estudiantes de doctorado y bastantes postdoctorados en el área de desarrollo de plantas. Y bueno, le agradezco mucho a Wolfgang, que aceptó inmediatamente la invitación y que compartió ya su conocimiento con los chicos de la licenciatura. Y bueno, hoy nos va a hablar de Decoding Root Growth to Address Climate Change. Gracias, Wolfgang.
Thank you very much. Muchas gracias. It's a pleasure to be here and to talk to you about the research that we have been doing in my laboratory at the Salk Institute to study root development and to think about how the study of these fundamental processes that relate to the genetic and molecular basis of root development and root growth and their responses to the environment might actually be leveraged to address the biggest crisis that we will face in the next decades, which is the climate crisis. And so I like to open my presentation with something that motivates me every day, and I think every plant scientist should think about, which is this very nimble graph. And this very simple graph actually outlines our future or our potential future on this planet, because it tells us what we have to do in order to limit global warming to a certain extent. So this is from the IPCC 6th assessment report. This is the International Panel on Climate Change that basically comes together every couple of years and solicits expert advice. And what they do is to take stock of the carbon emissions that we have and of the modeling that we can do to predict the climate change as it will happen. And so in this graph, you see the year here, and you basically see the gigaton emissions of CO2, gigaton CO2 equivalent per year. And all of these three curves are basically depicting scenarios that will need to happen to limit the global warming by the end of the century to a certain extent. So let's start with this one. This is, emitting, 55 plus gigatons of CO2 every year. That's what we're doing, globally every year now. And so if there's no change in terms of where our energy is gotten from, in terms of fossil fuels or and things, in the global economy, we are basically looking at 3.2 degrees Celsius increase of average temperatures on this planet by the end of the century, which doesn't sound a lot, but it is a lot. And everyone who works with plants, for instance, will know that during the summer, if we would increase the temperatures by 3 degrees, it will have remarkable negative consequences on the yield or things like pollination, for instance. So this is a pretty bad scenario, which I don't think anyone would like to have, with massive ocean rise and actually parts of our planet becoming uninhabitable, in particular in developing countries around the equator. Now, the hope of the global community, as you know, is to limit global warming by the end of the century to 1.5 degrees Celsius or 2 degrees Celsius, which are scenarios which still will lead to a lot of unfortunate change in terms of droughts and other catastrophic climatic events, but are still manageable, at least according to most experts. And you see what this will take. It will basically take to reduce our global greenhouse gas emissions from 55 gigatons of CO2 to around 20 when we want to limit this to 2 degrees Celsius, or even lower to around 11 if we want to limit this to 1.5. And that is a tremendous challenge because just reducing the CO2 emissions by 1 gigatons would require to basically change 200 million cars to electric cars. These are more cars than are on the road in the entire United States. So this is dramatic, right? So we have to think about ways how to actually, in addition, draw down carbon. So all these scenarios not only are only seen as realistic, but not only we stop burning fossil fuels pretty radically, but also we need to draw down carbon from the atmosphere. And so there we end up with plant biology. But you know, I think all these numbers, or like to the two main messages of this graph, relate very much to plant biology in my mind. One is it will get warmer, the climate will change, and get warmer and less you know, hospitable to agricultural production. So we actually need to climate adapt crops that they will be able to grow in a hotter climate and you know, experiencing more droughts, but also more flooding events. So that goes back to plant biology. Agriculture is actually a culprit in contributing to these CO2 emissions or greenhouse gas emissions, notably nitrous oxide. There's a lot of fertilizer being used, in particular in, you know, countries like the United States. And a lot of this fertilizer is basically being converted into nitrous oxide. It's not used by the plant, but converted by soil bacteria. And nitrous oxide is a worse climate gas, the greenhouse gas, than carbon dioxide. So there are ways how one can improve the ways how fertilizer is applied and how plants use it, and therefore reduce emissions. And the third aspect that I'm going to talk about and I'm working on quite a bit is that of plant-based carbon sequestration. I'm going to tell you in a minute why I believe this is a good and feasible idea and what we can do about it. And the carbon sequestration relates to this part. We have to basically not only reduce emissions, but we have to pull down carbon dioxide actively from the atmosphere, and there is no technical solution for that that is scalable. There are some technical solutions proposed, and there are pilots, but this costs thousands of dollars per ton CO2, which will not be feasible, unfortunately, to do on a large scale. So plant-based carbon sequestration might be a possibility to actually achieve carbon drawdown and storage on a large scale. Why do I say that? I say that partly because of this graph that actually convinced us that it's worthwhile to think about plant biology in that respect. So this is the Keeling curve. And I don't know how many of you have seen, you know, movies like The Inconvenient Truth from Al Gore, other like this. This is kind of the thing that everyone's worried about. This is the carbon dioxide concentration in our atmosphere, which was measured starting in the 60s on Hawaii by someone called Keeling at the Scripps Institute for Oceanography at UCSD. And this team measured every year. And what you can see is that 1960, if you were born 1960, the carbon dioxide concentration was around 300 ppm. Now it's 419 ppm. So this is like a tremendous increase of carbon dioxide, and this is what's causing the global warming. So, this is scary, right? But the good news is this: it doesn't go up monotonously, it actually oscillates. And so if you zoom in, you basically see that these oscillations are pretty remarkable. We're talking here about a multiple ppm change per year, which corresponds to many gigatons of CO2. And you can see when that happens, the lowest concentration each year of carbon dioxide is actually achieved in August, September. And the highest concentration is present in April, May. And that coincides with the growth season of plants in the Northern Hemisphere. And it basically is just like an indicator of the ability of plant ecosystems to make an impact on the global CO2 concentrations, because the reason why this is so low here, it's because, it was summer. So plants had absorbed a lot of CO2 throughout their growth season. And now in autumn, they start to, decompose in the Northern Hemisphere, and that then goes on until the spring of the next year. So that basically tells you that plant systems, it's an insight that plant systems have an amazing capacity to actually change the global carbon dioxide concentrations. Of course, we all know this because photosynthesis, the process by which plants fix carbon dioxide through the power of sunlight, right, make it into biomaterial, they remove that from the atmosphere, is very well known to us. and something that is you know, pretty remarkable. So the carbon cycle each year is on a global level, you know, modulate or basically determined by plants. And just to say that the amount of carbon that photosynthetic systems fix each year is basically many, many, many times more than all humans together emit through burning all the fossil fuels. So the idea is maybe we can actually engineer this carbon cycle by engineering plants or changing plants through breeding that somehow less carbon will be released at the end of the growth cycle. And of course, the vehicle for this will be the soil. So as you know, land plants have root systems, and these root systems grow in the soil. Every carbon that is deposited in the soil has a much more higher likelihood to remain longer in the soil than the carbon atoms that are fixed in the above ground tissue of plants. And accordingly, since plants have colonized the Earth's surfaces, the soils that have been built by the plant's activity contain now 2 to 3 times more carbon dioxide than the entire atmosphere. So the key would be to basically have plants that somehow deposit more carbon that lasts for longer in the soil. And so the next question would be, so if that could be possible, is there actually capacity in the soils to store even more carbon, to take some of the excess carbon from the atmosphere to keep it in the soil and thereby sequester it and keep it out of the atmosphere? And the answer to this is most likely yes, because we know that soils, you know, before humans invented agriculture, contained much, much more carbon than they do now. This is an example from a paper where a group modeled the loss of carbon that has occurred because humans started to practice agriculture and converted prairie land and forest and other lands into agricultural land. And in this process, soils already have lost an enormous amount of carbon, and they basically quantified, and this is over the time, they lost 133 gigatons of carbon, which is more than, there is almost 500 gigatons of CO2 equivalent. And so while this is, of course, you know, bad that, you know, humans have led to the release of a lot of carbon from the soils, it also tells us the soils have the capacity to hold much more carbon than they have now. So the idea would be, if you just had a method to backfill the carbon deficit in the soil, you could use it as carbon storage, and at least up to, you know, many 133 gigatons of pure carbon, which is a huge portion of what we need to draw down to limit global warming. Okay, so you know, inspired by these types of insights, at the Salk Institute, myself and these other colleagues, Joanne Corey, Julie Law, Joe Noel, Joe Ecker, and Todd Michael, got together over time and decided we really want to change our research to think specifically about this question, how we can understand the processes that can lead to more carbon storage and accumulation in the soil, and how could we engineer or design, you know, or inform breeding programs, for instance, to make this happen. And so we focused on 3 different traits that are pretty simple traits, and for which we had the evidence that actually a single gene or a couple of genes might be enough to enhance them. And one is more extensive roots. So every carbon atom that plants invest in their root system has a 5 times more likelihood to end up as a more stabilized carbon in the soil than shoot carbon. So it's really important to work on the roots. And so just by increasing root mass, we actually can generate a pool of more stable carbon in the soil and take out the carbon that is invested in the roots. And when I say that, I mean that is the carbon that it needs to build the roots, but also carbon that roots exude into the soil is already a win. The issue is, if you basically put all of this very shallowly, decomposition rates are pretty high in the upper parts of the soil. There's tillage. And so the carbon that is deposited very shallow in the soil gets quite easily lost. So a second trait that we are focusing on is to make these roots deeper, because with increasing root depth, the decomposition rate decreases. And in fact, if you look at the average age of carbon atoms in the soil, carbon in the first 30 centimeters of the soil have an average of, maybe a month to years, maybe a decade of lifetime in the soil. But if you go below 30 centimeters, this lifetime, the average age of the carbon that is in the deeper soil layers can extend to centuries or millennia. So the deeper carbon is deposited by soils, the more the likelihood that this will be long-lasting carbon. And the third one is to focus on the biochemistry of roots. Roots are composed of many different materials. They exude many different molecules. And some of these molecules and materials have a higher likelihood to be decomposed faster than others. And so we are focusing on biopolymers that are more recalcitrant, or where the literature indicates they are more recalcitrant. And our focus is on suberin, which you might know as cork. So cork consists out of suberin, which is a biopolymer that all plants make in their root system, is deposited in certain tissues of the roots, and is associated, or has been shown that in some soil environments it can be very, very long-lived, partly because it's highly reduced and can be degraded very, thermodynamically in an oxygen-poor environment. It's not very favorable to degrade it. Okay, so we have been working on this since 2019. At that point, when we got significant funding for these ideas, we decided we need to not only understand these processes, these traits. In 2019, we already had discovered a number of genes that can change these traits, but we really need to think about how could we make it more than just an academic project, but to bring it, perhaps facilitate to bring it out into the world. And thinking about how could this make an impact on carbon drawdown. And so our idea after many discussions was we need to use agriculture for this, because in order, if you would enhance plants by breeding or biotechnology, really the key is to cover millions of hectares with it, right? And so agriculture is already a system where 1.6 million hectares on this planet are dedicated to this endeavor, where seed genetic material is constantly updated. And so it's easy to imagine that you can bring in a trait that relates to carbon sequestration, and this could be distributed very, very broadly. And so we thought, in terms of choosing the species that we want to work on, in addition to the model species, and to translate what we find should be the species with the largest acreage, just because you get the biggest impact. And we chose 6 species: soybean, corn, wheat, sorghum, rice, and rapeseed. Now we have also some cover crops in it, because they were just a few species covering more than 50% of the arable land. So if it would be possible to improve these species and their ability to store more carbon for longer, you would make a big impact. All right. So that's the big picture. And so I'm going to tell you 2 stories about how we actually get at genes or genetic and molecular mechanisms that might be useful for engineer these traits, and that we are actually actively pursuing to engineer. And because our time is limited, I focus it today on the deep roots. We're working on all these traits. Deep roots are actually very interesting because for carbon sequestration, but also for a number of other things. Because with depth, if you have increased root depth, you can actually approach a couple of other climate-related problems that might be a win for the farmers. First of all, the question of nitrogen. So if you have deep root systems, you can actually intercept nitrogen that will leach over the growth season. So in the beginning, it's fertilized, and then with rain, it's leaching deeper into the soil column. If you have deeper roots and can absorb throughout the soil column, you will be able to intercept more of that nitrogen, which has really might have very large implications for trying to reduce this nitrous oxide emission that I talked to you about before. Also, water during drought is something. In rain-fed agriculture systems, if you have a drought, the water dries out from the top, so there's more moisture in the lower layers of the soil, and so a deeper root system will be able, for instance, during terminal drought, to assess excess moisture in the soil, droughts are predicted to increase in frequency. And then the carbon residence time, as I have been telling you, is also affected by deep rooting. So deep roots is great. So how do we get there? Yeah, and so you know, like all, at least of our discoveries, things start very small with our favorite model plant, Arabidopsis, which you see here. And in this case, we wanted to identify regulators that would allow plants to generate deeper roots. And something that is important to know is that the depth of root systems is determined partly and to a large extent actually by the steepness of the root system. So if the roots grow like very horizontally, they will not reach very deep. But if they grow much steeper, they will grow very steep. There's a major plant hormone called auxin that is known to be involved in determining the direction in which roots grow. And so what we did was to find a chemical inhibitor. This is like a well-established chemical inhibitor of auxin signaling. And we treated a couple of hundred different varieties of Arabidopsis with it. And so you can see that different strains, and I call them accessions, have a different ability to kind of, or a different sensitivity to this inhibition of auxin transport. You know, some don't change their growth angle dramatically, roots should grow down, but others get completely crazy and lose the sense, you know, the ability to grow along the vector of gravity. So we then use this sensitivity as a trait, as an input for a genome-wide association study, where we basically correlate over the genome scale the sensitivity to this NPA auxin inhibitor to genetic differences in the genome. And so we can do this over the whole genome. So basically every dot is a different position in the genome. And if there's basically a high value, that means there's a high probability that actually there's a genetic change in this part of the genome that is associated with the sensitivity of roots to this treatment. Okay, so we then basically focused on the most significant peak here on chromosome 5. And as you see here, this is basically a zoom in. It is in a typical Arabidopsis region where there are, where the genes are pretty dense, and it's in fact in the coding region of a gene here. So that basically then prompted us to ask, like in this whole region, is there any gene that is expressed in an area of the root where we know there is a function of controlling the direction of the root growth? And in fact, the gene in which the SNP was positioned that we had mapped using genome-wide association mapping was expressed in tissues of the root, and the expression of the gene is marked here in red in this very tip of the root, a structure called the root cap, and in the vasculature, in the elongation zone. These two zones are key zones or the key tissues that determine the ability of roots to grow in a certain direction. So that was a great candidate gene. So we then tested this hypothesis, whether this gene was actually involved in determining the sensitivity to this auxin transport inhibitor. And as you can see here, we generated overexpressors and a knockout CRISPR line compared to the control here. And when we exposed these plants to non-treatment condition, they all looked similar. But when we added this chemical inhibitor of auxin transport, the overexpressors as well as the CRISPR knockout mutants were more sensitive. You can see that here, where there's the statistical analysis, and that is showing us the root tip angle, so the offset of the vector of gravity. And you can see here that like multiple independent lines for overexpressing as well as knocking this gene out lead to hypersensitivity. So that told us, we are probably at the right candidate gene. The interesting thing with GWAS is not only that you find a gene, but you also find variants of a gene that are causal for a different behavior, right? So you're typically, you don't find knockouts or overexpressors, but you find often a kind of like a fine tuning of the trait. And so we wanted to test, if we were right, if we had identified the right genetic variant in our GWAS screen. And for this, we did this reciprocal transformation experiment where we used the knockout line, and then we used the variant from the hypersensitive lines to NPA and the variants from the tolerance, and we transformed it into this knockout plant. And the hypothesis was that if we basically complement this knockout with the NPA tolerant version, we should see a tolerant, NPA tolerant transgenic line. And if we did it with the sensitive gene variant, we should not see a complementation to this extent. That's what we did. In Colombia, which is the wild type, that's the positive, that's the control. We don't see a sensitivity to NPA. This is NPA, this is mock treatment. There is no statistical difference between these two treatments. In the knockout, as I've shown you before, there is a difference if you complement with the empty vector control. There is still a difference as expected. Now, if you complement with the NPA tolerant variant, we basically see there is a rescue of this sensitive phenotype because the complemented lines are statistically indistinguishable between mock and NPA control. But when you complement with the sensitive version, you don't rescue the phenotype, which tells us we really mapped the right variant. And that is very interesting because we can now dissect the molecular mechanism why these variants confer this different tolerance to NPA. But of course, you might have asked yourself, you're growing these plants on a drug that inhibits hormone transport. Does it have a real function for the control of anything that relates to root directional control? And so we started to test, conditions that we know tell us something about the tendency of the plants to grow downwards. And one very simple test is the ability to assess the gravitropism of roots. So normally, if you grow roots down, if you grow roots on a plate that is kind of vertical, they grow down. If you turn them on the side, roots will basically perceive this change of the vector of gravity in relation to their growth vector and start curving down. And so we tested whether the overexpressors or the knockouts for the gene that we had found were different in that response compared to the wild type. And so here, this is a time lapse. So you see on the left the wild type, and you see that after 2 to 4 hours, there is actually a correction of the vector of the growth, while the overexpressor is much slower and much more sluggish to do so. So you see it doesn't really, the wild type here is already completely corrected to the right vector, and this overexpressor in the gene that we had found is impaired in this. So we kind of did the statistical analysis for this, and you can see that when we look at the time it takes to correct the angle, the knockout line is affected in 2 independent knockout lines, as well as the overexpressor lines are affected as well. And that resembles really these different accessions when we measured this trait in these different accessions that had these different alleles of this gene. They resemble really one group of accessions that have one version of this gene that we had found, which is called the exocyst 70A3, is much faster in correcting the growth angle, while the other one is much more sluggish. So all being consistent. So the next question, and that's again important for engineering. If you really want to engineer a process, you have to understand it is how, why is that? What is this exo 70A3 gene actually doing? And so I want to take you back a step to this process of gravitropism, which is a pretty complex process, but the principle is simple. So if roots grow downwards, they basically have a symmetric distribution of this auxin hormone. So in green here, you see the distribution of auxin. But if they basically, if a change of the growth angle is happening, what happens is that this change in the vector of gravity is perceived in these root cap cells. They have very dense starch-filled organelles, and they push to on a different side of the cell as they would when they would grow downwards. And that leads to a reorientation of auxin transporters to only this side, and auxin is pumped massively to this side and not to this side. There's no symmetric distribution of auxin anymore. And auxin leads to an inhibition of cell elongation. And so if you ever blew up a balloon and put some tape on the lower side, you know that if you inhibit the extension on the lower side, but you have more extension on the other side, it will curve downwards. And that's essentially how roots change their growth angle. And so the gene that we found is part of this exocyst complex, which is preserved, conserved in animals and plants and yeast. And it's one of the 8 subunits, only that plants have very, very different isoforms for each of the subunits. And the important part about this is that the reorientation of auxin transporters that lead to this reorientation of the auxin stream is facilitated by endocytosis and exocytosis. And so, so we hypothesize that actually what happens if you have different alleles of this subunit of the exocyst, or if you knock it out or overexpress it, the rate by which auxin transporters are exocytosed in the membrane is changed. That changes the response to the gravity. And so we tested this. There are multiple auxin transporters in the root. They're called PIN transporters. And so luckily, the auxin field is very active, and we had a number of reporter lines already. And so we're looking here at the protein fusion. So you're looking here at the protein distribution. So in wild type, the PIN4 transporter is expressed in only a few cells, 1 to 2 cells here in the root cap. But when you overexpress the exocyst subunit that we had found, you actually get a mislocalization of the auxin of the PIN4 transporter protein, so that the expression domain is massively expanded. And I don't show the data here. Also, the reorientation upon a gravity stimulus isn't as quick in these lines. If you knock out the exo70A3 gene that we had found, you actually don't have any expression. You don't have any protein, PIN4 protein, in these cells anymore. But nicely, since it's expressed in the vasculature, we had the nice positive control that it's not just the microscopy. So whatever we do to the exo70A3, it changes the distribution of this PIN4 transporter. And we checked we checked at least 4 other PIN transporters, and none of them showed any change. So that's a very specific effect. And so we are kind of investigating now, how that actually works. It's pretty puzzling, but we know that actually PIN4 is downstream of exo70A3. Okay. So that was a very long dive in the molecular mechanism, but I can promise you that the more we know and understand on how these genes work, the better, the more engineering targets we have for engineering interesting traits. But of course, we didn't know whether actually these changes in these early processes and these responses to the gravitropism will affect anything that's interesting for deep roots, right? But we know that actually the direction at which roots grow will affect the depth of the root system quite remarkably. But in order to test whether it's actually true for our for the gene that we found, we had to develop like a new method, because Arabidopsis roots are very flimsy and it's very hard to detect them with non-invasive methods that you can use sometimes for other crop plants. And so we developed this pot chopping method. That's the postdoc at the project Takehiko. And the idea, or basically the principle, is you grow Arabidopsis plants in these large pots, and then you take a very sharp bandsaw and cut it through in the middle. You take basically a brush and clean the section, and then you take a high-resolution photo and mark up all root segments. And so if you marked up all root segments, you can actually quantify the distribution of the root segments per depth and measure root depth, or root depth distribution. And that's what we did here. Those are the 8 accessions in 2 groups. One group has one exocyst70A3 allele that we had mapped using the GWAS. The other group had the other exo70A3 allele. And what you can see immediately is that this group of accessions has a much shallower root system than this. So these kind of tend to grow steeper. So we basically then asked the question, is XO78A3, the gene that we had found, sufficient to actually confer this deep root configuration? And therefore, we basically tested our overexpressors and our knockouts. And as you can see, Columbia, which is the wild type, is pretty shallow normally. But if you just CRISPR out this gene, it's one base pair change in the whole genome, you basically switch these shallow roots to deep roots. If you overexpress it, you also change the root distribution to a deeper root system, but not as remarkably as through the CRISPR. And the downstream gene, PIN4, if you knock it out, you also get a switch. So just like that, and that's the statistic for that. And just like that, we had uncovered basically a regulator that can actually lead to a deeper root system. And so I will tell you later that how we are pursuing that. Okay, I'm going to skip that part because I'm running out of time. All right, so this is basically, and read the paper if you want to get the whole story. It was a long set, a very long list of experiments that we did. But essentially, in this study, we had uncovered really, the mechanism from the molecule to, the organismal phenotype with regards to deep rooting, and also uncovered some ideas about the potential adaptive benefit of these variants. Okay, so we are now actively pursuing to knock out orthologs of this gene into multiple crop species. A second story that I want to tell you is some kind of different approach, and that is using chemical genetics, and we are able to do some decent chemical genetics and GWAS on different chemicals with the help of Larry, which is the seed pipetting robot that we built, because we need to set up a lot of experiments with these tiny Arabidopsis seeds. And so using robotics actually helps us. And so we were able to actually screen through several thousand chemicals and to assess their impact on root system architecture, and then pick out interesting chemicals that might lead to changes, or that lead to changes that might affect root depth. And so I'm just going to show you one story about these. One of these chemicals is called mebendazole, and it led to a very striking change in the angles of these lateral roots compared to the non-treated control, which was it made the roots grow much more lateral. Now this, you might think, is not helpful for getting deep roots, but it is helpful to uncover the regulatory mechanisms that actually determine the set point angle, is what we call it, the angle at which the roots grow. So we decided to try to understand which genetic and molecular pathway this chemical interferes with. And so, so the key to this was to basically first, chemical genetics is a little bit tricky, because if you apply a chemical to something and you let it work for a while, you might actually, and then you try to do RNA-seq, for instance, you might catch a lot of indirect effects, but not really the culprit that is really acting on. And so in order to get as close as possible to the molecular target, we actually did a time course where we put plants on MBZ plates from normal conditions, and then did time course and watched when do the first changes occur. And you can see here, after 2 hours and 3 hours, the first changes occur to these root tips, and they start growing more to the horizontal position. And you can also do the reverse experiment and then basically quantify when do the changes occur. And that would inform you on when to do an RNA-seq experiment to see which pathways are actually already affected very early. So we took this 3-hour time course and we did an RNA-seq, and to our surprise, there was a clear winner of an enriched pathway, which was the negative regulation of ethylene activated gene expression. Ethylene is another plant hormone that hadn't been implicated with lateral root angles or the steepness of the root system before. So we wanted to be extra careful to see whether that's actually involved. And so we did a lot of, we did some time-lapse imaging to make sure to rule out other hormonal pathways. by basically treating with MBC and with distinct hormones and looking at reporters. And we found that actually the major response that was elicited already after 20 hours, after 20 minutes, was the response to of the ethylene pathway, and not the others. So the first step to get to the target was to ask the question, where does it act? So the first hypothesis was this chemical binds to something that produces ethylene or is itself converted into ethylene. And so we measured the ethylene production. This is our positive control, ACC. It's a precursor of ethylene. And this is basically the concentration of ethylene we detect. And we can see that while the precursor of ethylene, when you put it on plants, leads to like a massive increase of ethylene concentrations, MBZ doesn't do anything. So it's somewhere in the signaling cascade of ethylene. And so for this, we basically went down the known pathway, and we always ask the question, are mutants that are affected in ethylene perception or signaling insensitive, right? Or are they not insensitive? And that way we would get to the molecular target. So the first step in ethylene perception is the receptor ETR1, and when we basically treated ETR1 with our MBZ treatment, which is now labeled differently, but this is MBZ, we actually elicit basically the same response as in Columbia 0, which are the wild type, which basically tells us it is somewhere downstream of this ethylene receptor, right? Because we can still elicit this response. So then the next step in the pathway was the CTR1 protein kinase, which is involved in the signaling interaction and signaling transduction, because it's phosphorylates components. And so there we could see that actually the CTR1, which are kind of messed up developmentally, but importantly, they don't respond to the MBZ treatment compared to wild type. So that told us it's very likely that MBZ acts actually through CTR1. And so, so then the next one downstream of CTR1, the next signaling component is IN2, and it was also insensitive to our SMZ treatment. And so that tells us it's most likely CTR1 because it's the most upstream component that's insensitive when you mutate it. So to get additional insight, or to test it from a different way, we actually were very fortunate as MBZ was already being described to have an effect in human cells on cancer. And so people already had isolated a crystal structure in human. And in human, it basically, MBZ is an inhibitor of the MAPK kinase 14, and the structure was resolved. And essentially the key amino acids by which MBZ is bound in the human system are conserved in the CTR1 kinase, which is pretty cool. You can also do the molecular, docking analysis, and again, it basically tells us there is a great fit of MBZ into the active domain of CTR1. Okay, I'm going to skip that. So basically, we through this study we had uncovered a chemical that basically activates the ethylene pathway, and through this changes the root angle. And so that was pretty cool because ethylene was not implicated in this process. And now we have a whole different target that we can use to try to engineer differently shaped root systems. And the beauty of this pathway is that it's very well described. So we have a number of highly conserved gene targets in different crop species through this. Now you might say, okay, you just showed us that you can make more shallow roots, but not deeper roots, because MBZ had this, shallow root effect. So we thought, oh, maybe let's revisit our data to see if in negative regulators of this process, we actually see a changed root system that would be steeper. And so when we actually went back to our data, we realized that the IN2 mutant, which is actually doing the opposite to MBZ, actually does have significantly different angles, so you can see. And so I'm going to keep you posted. Okay, so the last couple of minutes, I will show you a little bit what we do with real crops. And the way we are basically leveraging what we have learned and what we can do in different ways. One way, as I was telling you, is that we actually CRISPR engineer or we overexpress genes that we find in Arabidopsis and that are conserved. And we're doing a lot of work to enable us to do that, because finding the right ortholog in crop species compared to Arabidopsis is not trivial often, because plants are amazing of shrinking and expanding gene families. Is that a signal? Okay. Good. Yeah, so expanding gene families, but we're using a lot of single cell RNA sequencing to actually find orthologs that are actually expressed in the same cell layers like in Arabidopsis. So basically homology plus expression location, in our opinion, will give us actually a very precise way to predict the right targets. Another thing what we do is to do something like this. This is our greenhouse team. We have built a 10,000 square feet greenhouse 10 miles up the coast, where we actually do a lot of crop growth, and we grow diversity collections of crops. Like this is, for instance, our soybean cylinder experiment, where we grow, where we have grown like almost 1,000 soybean lines in these transparent cylinders. And we have engineered a system with which we can actually take rotational series of the root structures so that we can quantify the root architecture in 3D and measure the steepness of the roots, also the growth and other features. And those is an example of two different soybean accessions that are very different in terms of their biomass and their tendency to grow deep. And so with this data, we can also do genome-wide association mapping crop species. We've done that in multiple species now. This is an example for the low tip depth, where we basically map how deep do the tips of a root system go in this cylinder. And so we can basically try to identify the genetic variants that are associated with this. You can also identify soybean varieties that are already pretty good at forming a deep root system, and then test whether it actually holds up in the field. And one of the cool things about these GWAS, in particular this GWAS, is that one of these significantly associated SNPs was also an exo 70 subunit. And so we got very excited, because that was the same gene family that we had identified in Arabidopsis. And we thought, before we do, in crop species, making CRISPR takes a lot of time and effort. So is there another kind of data that we can use to get more confidence in this hypothesis? And one is actually the single cell sequencing, where we ask, would like genes that we think might do the same thing, are they expressed in the same manner? And one thing about this exo 78.3 in Arabidopsis was that it was expressed only in 20 cells out of 110,000 cells in the root. It was only expressed in very few cells in this root tip. That if you get the expression profiles of all the cells in the root, you just get a handful. And so this one that we mapped in soybean, and that's our soybean data set, and each dot is a nucleus, was expressed in 2 cells out of the 30,000 cells that we have mapped. So again, another indication that this exo 70 subunit, just like in Arabidopsis, is expressed very, very specifically. And so we are very confident that this actually might be a subunit that might have a similar function in soybean. And so we are knocking it out right now. And so in a year, I hopefully could tell you more, because it takes a while in crops. Another thing that we do is, and that's really the last slide before the music kicks in, is that we test hypotheses in the field. This is, so the issue with field studies that are coming from being a lab biologist is that, you only have one growth season usually. And so that doesn't give you a lot of shots on goal. And so in order to get more, we're actually doing experiments in the US, but also in the Southern Hemisphere. And so we set up an experiment in Chile in 2021, in the winter of 2021, well, the summer in Chile in 2021, where we basically planted soybean varieties in the field, and we used a number of tricks to measure their roots. One was these electronic devices called the root trackers that can measure in real time, the steepness of the root system early on. But we also did like some more, traditional thing like hammering cores in the soil and pulling out the roots, washing them, getting a good luck cat. No, I'm kidding. Washing the roots and basically testing the root depth distribution in the field. For soybean varieties that we had determined in the greenhouse, they should have a different root system architecture. And just like, the issue with plants is there's a huge environmental component for shaping the root system. And so we really want to make sure that what we observe in our greenhouse in California holds true in fields, because we want to, map the right traits. And so we basically harvested all the roots with these cores and also these crown roots of the soybeans, and then basically wanted to test whether the extreme contrasting accessions from soybean that we had identified in California would still be different in the field. And that's, one result where basically, a result that made us very happy, because these two contrasting accessions of soybean that we had planted, and those were 2 out of 24, they showed a difference in the gel and in the greenhouse, but also a difference in the field. So basically, these were much more massive in the gels and much more steeper oriented, and that held true from these crown roots that look very different from those. So we think we have a good predictive ability to, at least to some extent, to use the data and the genetic data from the greenhouse. And that's also another data point, very preliminary, but I'm very excited about this, where we basically take, the angles that we measured in the greenhouse, and we ask, is that correlated to the root mass across the depth from the soil cores from Chile? That's data from California. That's data from the field in Chile, and there's a very high correlation. But of course, it's just one data point, 1 year, one season, and it's not very many lines that we took, but it's very encouraging, because we think we might be meaningfully measure these traits and then evaluate engineered varieties, for instance. Okay, with this, I want to close. I want to thank the scientists in my lab who did the work that I presented. Those were all very talented postdocs who did different things. Takehiko worked on the EXO, Wenrong and Hai-An worked on the MBZ part. Ashish works on the field studies. Charlotte did the single cell experiments, and Ling is our bioinformatician magician. And then my colleagues with whom I acquired all this great funding, and we all do different lines of research in our labs that, we want to lead to the same aim, to understand mechanisms and engineer mechanisms to store more carbon for longer in roots. And with that, I'm going to take any question.
Preguntas.
Very nice talk. Do you know if changing root architecture would affect yields of the plants of the crops you selected?
Yeah, so that's an excellent question, and that's something that we keep in mind. Clearly, if it is you want to have the same yield if you want to have an engineered variety, and so the answer is complicated because it depends on the crop. Like each crop has a different physiology. Wheat is very different from corn, and soybean is very different from wheat, and so on and so on. And so I think the extent to which you can change, for instance, root mass and root depth without affecting yield might be different. Another factor that is very important is where are these crops grown? Like if you grow a crop in a rain-fed environment that experiences drought, a steep root system architecture might actually provide you with higher yield in some species. And so I guess the answer is it depends. There are many, when you look at many varieties in different plant species, also at the distribution of root systems in ecological in ecological studies, there doesn't seem to be a very clear trade-off. So there is some wiggle room, but really the details will be very important. Because the key is, what is the limiting factor for the plant, right? If water is the limiting factor for the plant yield, right, you can easily imagine that actually a steeper root system architecture might improve the yield, and you might actually allocate more carbon to the root and get the same yield or better yield. If sunlight or carbon is limiting, then of course you would face a zero-sum game, and then you would expect trade-offs.
Yeah, thank you for your nice talk. Very interesting data. And I'm wondering first, in relation to the big picture, what did you expect from the plant metabolism in the shoot? You didn't tell anything about what is going on with the shoot functions when you have, as you just mentioned now, when you have a peak of a metabolism using CO2 in the root, no? So what is your thought about that? That's one question. And maybe I can tell you the other question afterwards.
Absolutely, yeah. So as I was saying, I think there are multiple I think considerations that relate to the shoot. So one consideration would be the question that I just discussed, which is, would there be a trade-off? Is there a limited carbon budget in plants? And that would an increase of the allocation to roots decrease the yield? And I think, again, like this depends. I think there are good examples like the DRO1 in rice, for instance, where actually under drought conditions, you achieve much better yield with a DRO1, allele containing plant than with a not DRO1 containing alleles, and there are other examples. The second consideration is there are many groups in the US, for instance, that work on improved photosynthesis, like for instance, engineering photorespiration, the group, the RIE project in Illinois and so on. So that could be additional to enhanced root function. And so I would expect there would be at least an additive effect. So it would be, if you engineer the root to basically get more water for longer in the growth season, and you engineer photosynthesis, bringing it up closer to its theoretical limit, then you might actually get, like a win-win situation. And so we are actually trying to find funds to work with the photosynthesis engineering people. So, and the third consideration relates to the type of engineering you do. So, because one factor is not only the carbon allocation to build the root structure, another factor is the respiratory activity. Root respiration can make up to 30% or even slightly more of the total carbon expenditure in plants. And so if we think about deep roots, if you don't increase the root mass, there's no strong expectation that respiration will increase. If you make more roots, respiration will increase. If you make more suberin, that is not necessarily a strong effect on the respiration, because suberin is basically dead. So you don't have a higher kind of steady state metabolic cost for forming more suberin, just the carbon that you invest in the suberin itself. It's the same when people think about erynkema, which is like cheap to produce because it's essentially not metabolic active. So there are a number of, different interventions have different effect.
Yeah, well, okay. We can discuss. I have more questions about it, but the other thing that I wanted to ask you is, in the plan, in the Arabidopsis XO783, you look at the shoots, you
Yes.
look at the biomass in shoots, and you look at the harvest index in Arabidopsis.
We looked at the seed count, which is similar. So there was no difference in any of the traits that we looked at under regular conditions, so under normal growth conditions. So, but there is a caveat, which is these plants were grown in the lab, right, and under very controlled, very happy conditions. So if you would grow them in a field, there might be, a different effect. Another thing I didn't present it here, we have identified several genes that affect root biomass dramatically. So we have a number of genes where we can increase the root biomass by 200%. And in these plants, Arabidopsis, in the lab, we also get like a 150% increase in shoot biomass. But again, this is like perfect conditions, right? And so you might actually buy, if you have more growth, there might be, other risks, disease susceptibility or anything else that might occur. But it is possible to increase biomass of root and shoot at the same time dramatically.
Yeah, it may be, yeah. So you said that this defect in the inoxin transport is not affected by stress, by any kind of stress. Did you say that? I mean, you test your variants in different drought or?
Well, we tested drought. I skipped over that. We tested drought conditions, moderate drought conditions, and in that case, basically there was a difference. So under drought conditions, the root system architecture has a strong effect on a number of seeds.
Oh, okay.
Yeah.
Okay. Okay. That's it for now. Thank you. Thank you very much.
¿Alguna otra pregunta aquí?
Thank you. I just want to ask you, like in the case when you treat the plants with the MBS, so do they change the growth in any direction, or it's always going up like agravitrophic?
Yeah, so no. So in the case of MBZ, it really changes the set point to a more horizontal. It doesn't lose the sense of direction at all. So and that is, one of the interesting things, that it changes the set point. So when you have the different roots, like for instance, primary roots, they have a set point along the vector of gravity.
Right.
And then in Arabidopsis, lateral roots, the first order lateral roots have like a set point around, like 45 degrees first. And the chemical set the set point to basically 90 degrees.
Right. Because sometimes I can see that it's growing like that with the 45 degree, and then it's like going a little bit up.
Yeah. So it's a little, but it does, like an average, if you look at the, at the average effect is not, it's not.
Okay. So it is my impression that when you have all those mutants, like the XO78-3 or the treated with the drug, you do see the deeper root, but do you see like more lateral root on the top part? So does it have in the lateral root an effect on the lateral root development?
Yeah, so for the the exo, like the genetic, like the overexpressor or the knockout mutant that didn't have any difference in the lateral root density.
Right.
That we observed. We didn't check for lateral root, primordial formation. Like we didn't check that.
Okay.
With the MBZ, I don't think we really were able to assess that in a meaningful way, because MBZ also has other effects on the primary root that make it then more difficult to interpret the results. So, so in that case, also ethylene, or if you look at the mutants, ethylene has a number of effects. And so that is why we are basically engineering, for instance, the ethylene genes in a cell type specific fashion.
So in the case of the soybean, the mutants, that is mapped also very close to the XO78. So do you see an increase of the root architecture? Have you looked at the nodulation, for instance?
Not yet.
Could you increase the nodule development? That could be nice to see.
That's a good question, but we haven't assessed it yet. But with more, in particular, when we have like a clean knockout, then we will be able to look at the nodulation.
So just a short question is, you have a change with the MBZ with the lateral roots and the angled. How do you try to look at the root hairs, because those are very set up also for an angle?
Yeah. So if you treat longer with MBZ, you get like a crazy amount of root hairs. So, so yeah, and that's consistent with ethylene having like a number of effects on, on root development.
We have two questions by Zoom. The first one is by Dr. Adriana Garay. She asks, do you know if it is more important to change the cell proliferation or the cell elongation to achieve the longer root?
Well, that is a great question. I think, we haven't like done an experiment to address that, but you need, you can elongate cells only that much. So if you don't have a constant supply of new cells, you're limited. So my answer would be if for instance we're looking at accessions that are very fast growing, you usually have increased cell production as well as cell elongation. So in order to get the longest root, I would say you should increase both. And in fact, many of the developmental genes that are known to lead to a longer root change both.
Okay, and Adriana also asked, along with the previous question, what happened with the meristem cell number and the length of the completely elongated cells in the XO738 mutant?
So we haven't quantified, like other than basically determining whether the meristem looks different, we haven't quantified changes. There were no apparent differences between the mutant and the wild type. Okay. But we, there might be something that is not like very obvious.
So Berenice Garcia asks, the phenotype with the MBC inhibitor is strictly similar to the HY5 mutant. Do you know if there is a crosstalk between CTR and H5?
H5?
Yeah, H5.
Yeah, I don't actually know, but there might be. Yeah, because H5 is definitely regulated in controlling root growth and lateral root traits. So there might be a crosstalk, but we haven't looked at it.
Is there another question? I have one quick one. So the amino acid that changes by the SNP, does it prevent interaction with the XO7A with the downstream molecule?
So far, we haven't tested each individual change. So there are a number of amino acid changes that occur. So we don't. So far, we haven't been able to assess that. And so there are also a number of SNPs in the promoter. So it could also be related to the expression strength, for instance. Yeah.
So if there are no more questions, thank you very much, Wolfgang, again for your presentation.